So in terms of the next generation sequencing data analysis, what we are looking at is, for example, these are results, and this is a very useful link. It teaches us various tools that allows us to interpret the results. So there's something called as SAM-BAM, and SAM is the alignment is an alignment report in a sequence alignment report that is that is kind of human readable, and binary BAM is which is machine readable. So we are going to look at some aspects of this, not all, but some that are kind of crucial. So, for example, if you look at the first field here, and if you look at the first line here, each of this is explained. Each of this is explained here, and for some of these, you can look at this resource, and it gives it better explanation. And once again, right now we don't have any data, but if you are working with next generation sequencing data, some of the details of these results become, it's easier to understand when you actually have numbers to work with. So I'll talk about a few of these. So the first field that is here is the same as what is called the Q name. Now remember, at, for example, FASTQ, you see that the at followed by the sequence ID tells you a lot of this information. It's necessarily going to be read, but it tells you more information about that particular read. So this Q name, this first part, is related to the information that comes after the at. Okay, so a lot of the information is built in. It's important more from the perspective of understanding what kind of information is here. The second one is what is called the flag, and the flag has to do with particular codes, and they have to do with whether the reverse sequence is aligned with the reference, whether the forward sequence is aligned with the sequence, etc. Again, this particular tool will give more information about this. The position is the starting position of the read with respect to the reference sequence. Map Q is related to the score. The log inverse of 37, negative log inverse of 37, it would give you the kind of the Phred score. Cigar we will look at in the next slide, and 37M just means that all 37 nucleotides associated with the read match with the sequence, and that's basically it. Can get a lot more complicated, and so we can count here 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37. So that means all 37 match to the reference. For example, and you can see the sequence here, that's the read. And we look at the qualitative and the quality scores. And remember, the quality scores are the ones we looked at here, right? The ones we looked at here. So for example, it tells you kind of a qualitative notion of exactly what that score is, what the confidence in that particular nucleotide being correctly assigned to that particular position is. And that's all we look at. If you look at this data analysis, we'll see what is called the CIGAR score. Now, let's say this is a reference sequence. Again, so this does not work in de novo, okay, in de novo assembly of genomes and large sequences. And this is your read. And so what we have to do then is we have to align this with the reference sequence, right? And that's kind of the point of you take your sample sequence and then assign it and kind of stitch it together. And so these are the positions. And then you align the two. And what you see in this alignment is the alignment actually begins from here, ACT, ACT, ACT. There is an insertion here, then the next three are matching, then there is a gap here, which is a deletion, and then these match. So it's a pretty good match. So the question is, what information do you get related to this match? So here's a score. This is a CIGAR score. POS, position 5, and the CIGAR score is 3M. Now remember, in the previous example, we saw that the CIGAR score was 37M. So in this case, what you'll see in terms of the CIGAR score is that 3M, first of all, position 5, which means that the alignment against the reference, just like we looked at in the last slide, occurs at position 5. And then you see 3M. 3M means that three of the residues match. Okay, three of the residues match. Then there is a 1I. That means that there is an insertion. I stands for insertion, and there's one nucleotide. I keep saying residue. It means nucleotide. One of the nucleotide is inserted. Then we have three matches, 1, 2, 3. Then we have to put a gap in here so that the rest of the sequence can match, and that for is in the case of our read a deletion. And then the next is 5M. 5 of the nucleotides in that sequence are matching. Okay, and that's why the score. Now you can look at, so those were typically matches, insertion, deletions, and then so there's a whole bunch of other things that are involved, that if you looked at certain reads, you would be able to make a cigar qualitative score out of this. So you see that this is the bits. So if, for example, this is the code for kind of human readable, and this is the code for machine readable. M for match, I for insertion, D for deletion, etc. And here's a pretty good rule, as samv1.pdf would be a good resource which explains some of this. Now, another kind of a statistical metric that you see is what is called the N50 and L50. So let's think of what the Broad Institute defines the N50 as. So the N50 is a weighted median. Is defined as, so for example, we're looking at these contiguous sequences called as contigs. And if you collect all the contigs of a particular length, what is the N50 length? The N50 length is a median, but it's not quite a median. You remember from any discussion of median, and those of you who heard this, who listened to the lecture, especially in class on statistics, on kind of the introductory statistics that we looked at, what you realize is the median is, you place your observables or your measurements in ascending or descending order. The middlemost number is your median. In this case, it's a bit of a weighted median, which is the N50. So if you look at all the kilobase pairs in a particular contig, if, for example, the contigs that contain more than half the sum, then that particular contig length is called the N50 length. Okay, it's a weighted median, and the number of contigs which give us an N50 is called L50. Let's look at the next example, an example of N50, and it comes straight out of Wikipedia, and you can look at several of these. Now, so we have 6 contiguous sequences. One is of length 80,000 base pairs, and 70,000, 60,000, 50,000, 30,000. So you're adding all the kilobase pairs. Okay, and that gives us 290,000 kilobase pairs. Now, how many of these contiguous sequences result in more? Now, if we had, for example, just placed these 5 numbers in, the median would be at 60. But because the number of kilobase pairs is 290, if we line up all the base pairs, you realize that these two numbers, 80 and 70, give you 150 kilobase pairs. So 150 kilobase pairs is more than half of the total base pairs count, which is 290. And therefore, that half, that, for example, that particular median number comes not in the 60 kilobase pairs contig, but in the 70 kilobase pairs contig. And that is why N50 is 70. Okay, so when you assemble all the contigs in a line, the middlemost number comes in the contig that is 70 kilobase pairs long. Now, how many contigs are required which constitute the N50, the 80, and the 70? Because the 80,000 and 70,000 kilobase pairs make up more than half of the total number of base pairs, right? And so the number of contigs in this case is 2, and that explains the how. Here is a very good resource which is used in next generation sequence analysis tools, and there are many of this kind, and that is something that you want to understand. Now, Velvet is something we will look at a little bit in terms of of the how this assembly takes place. Here's some more software that you could that you can use, different resources. The Broad Institute has the GATK, and you can download all these things, and it feeds into, you feed your software into this, and it gives you the results. It does the assemblies, it puts the reads together. There's different kinds of resources. Let's go back and look at what is called the Tuxedo Suite. The Tuxedo Suite is consists of bow tie, top hat, and cufflinks, and these are literally software which actually take the sequence, and they kind of, you can script them to the results of one are the input to the other, etc., and you can kind of script them to run all together. Okay.